Good evening, everyone. I'm going to tell you a story about a set of twins. And most of you will know that there are two types of twins. There are fraternal twins and there are identical twins. And fraternal twins come when two separate eggs are fertilized. And so you have two siblings who share 50% of their DNA, but are otherwise not identical. Of course, they're more similar than other siblings because they share an intrauterine environment. Identical twins, or what we call monozygotic twins, in contrast, are born when a single ovum is fertilized and then splits into two separate embryos. So here, for example, the different types of identical twins that we can have. On the left there, we have dichorionic diamniotic twins, where the ovum splits early at the moral stage within 4 days of gestation, and you have two babies who have separate amniotic sacs, and then the next one along we have two separate placentae as well. The third one along there is the monochorionic diamniotic twin, and that's what I'm going to talk about today. And in this case, the early blastocyst splits in the first week of gestation before it is implanted in the uterine lining. Then there's also monochorionic monoamniotic twins, in which the developing blastocyst splits even later, after implantation. And here you can see that the two babies share the same amniotic sac, so they're intimately intertwined in the uterus. And then missing from here is the type of identical twin that occurs when the embryo splits even later, and that's the Siamese twin or conjoined twins. Now, these two types of twins here, the monochorionic twins, are very high-risk pregnancies. They're high-risk pregnancies because the fetuses share an intrauterine placenta, they share vascular connections through the placenta, and obviously in the case of the one on the right, the monoamniotic twins, they actually share space and therefore can become entangled. The twins I'm going to talk to you about, and I have permission from the family to talk to you about these twins. I also have permission to show photographs. I'm not going to do that because the story is rather a sad story, and some might find the photographs a little bit distressing, so I'm just going to focus on the story instead. These twins, the pregnancy went well for the first half of the pregnancy, but then at the anatomy scan, around about 20 weeks, the finding of a double bubble was identified in both of these twins. Now, a double bubble is a characteristic ultrasonographic finding, as shown here, where you have a dilated stomach and also a dilated duodenum, the first part of the intestine. And this is a classical sign that suggests that there is a blockage in the developing intestine. Now, of course, the fetus in the uterus actually swallows a lot of amniotic fluid. Starts swallowing at about 11 weeks gestation, and by 36 weeks gestation is actually swallowing about 3/4 of a litre a day. So per kilo body weight, that'd be the same as you drinking 14 litres a day. And so when the fetus is swallowing that amniotic fluid, but the intestine is blocked, the intestine proximal to that, the duodenum, the stomach, become very dilated, and that's what we see here. Both twins also had what we call soft dysmorphology features. By this we mean features that are how they appear, but they're soft because they don't have pathognomonic characteristics. That is, they're not diagnostic. They just are particular features of these babies. In this case, they have brachycephaly, which is flattening of the back of the skull. They had a flattened facial profile, particularly in the mid-face, and they had short femora or thigh bones. Now, these sorts of features can occur in normal people as well, and it's often the constellation of these together can form a pattern. Now, unfortunately, in this pregnancy, in twin 1, the condition progressed to something we call hydrops. And hydrops means that there's an accumulation of fluid in more than one body cavity in the fetus. In this case, it was in the chest cavity, between the lung and the chest wall, and in the skin, what we call edema. Now, hydrops in a fetus is a very serious condition. It can be due to multiple causes, but it can often be fatal in pregnancy. Now, the problem, as I've mentioned already, is that monochorionic twins are very high-risk twins because they share vascular connections through the placenta. And so if one twin is at significant risk of demise, then obviously that's terribly sad for that twin. But if the twin should demise in utero, that then can have significant consequences for the surviving twin because of the vascular connections, meaning that a sudden drop in blood pressure in one can have major consequences for the other. And so when the obstetricians became anxious that this twin was at significant risk of demise in utero, an emergency cesarean section was performed at just under 30 weeks. So this is 10 weeks early. Now this twin that had the hydrops had severe generalized edema or fluid in the skin, and unfortunately died an hour after birth. And it was found that this baby had very small, underdeveloped lungs, pulmonary hypoplasia, and also did indeed have a blockage of the small bowel. And this blockage was in the jejunum, which is the second part of the small bowel, and I'll show you a diagram of that in a moment. Now the other twin was born without any evidence of hydrops, but required respiratory support and was intubated only with exceptional difficulty. Now remember, these are identical twins, so they're genetically completely identical, where DNA is completely identical. And yet here they have different characteristics. Both twins did have these soft dysmorphological features after birth, the flattened bridge of the nose here, a nasal tip that sort of tips up, a tented upper lip, and the brachycephaly that I mentioned earlier. So twin 2, after birth, this is the radiograph after birth, and you can see this is the postnatal equivalent of the ultrasound that we saw before. So here you have a very distended stomach, and here you have the distended duodenum, and then a distended proximal small bowel. And when this baby had surgery to address this blockage, was found to have a blockage in the jejunum also. A complete blockage, and in this case, a particular rare type of blockage of the jejunum called an apple peel bowel anomaly. And I'll tell you a little bit more about that in a moment. And these apple peel bowel anomalies have a very unusual blood supply that is quite precarious. The baby also had a malrotated obstructive cecum. The cecum is the first part of your large bowel from which the appendix dangles. And normally if you have a malrotation, the surgeon will try and correct it and put it back in its correct place so that it doesn't twist at a later time. But when the surgeon tried to correct this malrotation, that precarious blood supply I mentioned was blocked off, and the apple peel segment became quite ischemic. So the surgeon couldn't proceed with that and had to leave it in its malrotated state. Now interestingly, this baby also had other features that its genetically identical twin did not have. This baby had laryngeal dysgenesis, that is, the vocal cords were malformed, underdeveloped, leading to what we call a critical airway, which means that the tube through which the baby breathed was very, very narrow, making it extremely difficult for the baby to breathe. Also had high blood sugar levels, and also had a ventricular septal defect, which is the common type of the hole in the heart, that can be anything from a fairly insignificant in a normal healthy baby to a more significant defect that requires surgical closure. So this is a diagram of your intestinal systems. Here you can see the stomach and the duodenum, and then this is the jejunum here, and the blockage in these babies was in the first part of the jejunum, so hidden behind the colon here. Then you have the terminal part of the small intestinal ileum, which drains into the cecum. So this is the part that was malrotated, meaning it was in the wrong position, it was twisted round, your appendix, and then your large bowel. Now I mentioned that this baby had a blockage in the jejunum. There are these 5 different types of blockage in the jejunum, from a simple web, so that there's continuity of the bowel wall, but a blockage internally, through to a blockage with a bit missing, a complete gap, and then this is the apple peel bowel. And I think you can see why it's called an apple peel bowel, because the bowel is twisted in that way that if you try and get the apple peel off in one go without breaking it and then hold it up, that's what it looks like. And here you can see the precarious blood supply. You can see why that's so precarious. It doesn't have any tissue supporting it, whereas normally your blood supply lies within this mesentery that gives it support and maintains it in the correct position. And then the final type is when you have multiple blockages all the way along. So here we have a diagnostic dilemma. We have genetically identical twins, but they have different congenital anomalies. They're phenotypically different. The surviving twin has multiple complex, life-threatening anomalies, but no diagnosis. To have a chance of survival, this baby would need several things addressing. First, the laryngeal atresia or laryngeal dysgenesis was so severe that the baby was going to require a tracheostomy, that is a breathing tube through here, and that would require reconstruction later, meaning the baby is likely to be in hospital for at least a year and possibly more than that, even 2 years sometimes. Secondly, we have this bowel that we know has a precarious blood supply, and should that bowel twist again, as it has done already, that could lead to necrosis of the bowel in a critical situation that either requires surgery or is fatal. So prognostication and counselling is therefore extremely challenging here. We don't know what the cause of the abnormalities are. We know that there are major abnormalities that could be life-threatening, but we don't know what the outcome of this baby would be were we able to fix those abnormalities, because we don't know what the underlying cause is. Now, the standard approach to this sort of baby would be to undertake a hierarchy of investigations. You start simple and you gradually build up to more complex investigations. And indeed, that's what happened in this case. So you start, for example, with baby had what we call a karyotype, which is a genetic test that's been around a very long time, and that was completely normal. Then have what we call a microarray, which is the sort of slightly newer, now still quite old, but a newer test that tests for certain gene mutations. That was also normal. Had imaging to see whether the pancreas was normal with that high blood sugar. Had numerous blood tests. Had lots of specialists come in, none of which led to any diagnosis. Now, there is available internationally an alternative test, which, and by all of this investigation took at least 2 weeks by this stage, by which time we were no further forward. And internationally, there is an alternative test called Rapid Proband Parents Trio Whole Genome Sequencing with phenotype-driven gene prioritized data analysis, which can give you the answer in a very rapid period of time. For example, in the US, they are now doing this getting a result in 12 hours. In Australia, in Sydney and Victoria, it's available with about a 72-hour turnaround time. It's not available in New Zealand yet. So we decided, and what this means, the Rapid Proband Parents Trio Whole Genome, what that means is the proband is your case, the baby. The parents, that speaks for itself, both parents, that's why it's a trio, because you have the baby and both parents. Although in this case, of course, we had 4, because we had twins. Whole genome means you sequence the whole genome, not just the parts of the genome that make up your genes, but all the bits in between as well. And then phenotype-driven means that you take into account the phenotype of the baby, the characteristics of the baby, and you match that with what you find in the genotype to see whether there's an abnormality that you can find that is likely to explain the problem. So this is, for New Zealand, it's quite complex because we have to send genetic material overseas. It's costly, it costs about 18,000 Australian dollars, and there are lots of approvals to go through. However, in this case, we did get approval, and to the credit of the clinicians and the managers at the hospital who needed to give approval, they did it within a few hours. It took a little bit longer to get the samples over to Australia, unfortunately, due to the issues of sending material offshore, but we did get the results back in a pretty prompt period of time. And indeed, this did find a mutation in these babies that was not present in the parents. And this was a de novo, the new missense variant in a gene called ACTB, which is a beta-actin gene. And what this means here is that at position 1043 in the genetic sequence, the cytosine has been changed and you get to a thymine, and this results in a single amino acid change from a position 348 from a serine amino acid to a leucine amino acid. This is your beta-actin protein. This is where the abnormality is. So one amino acid change just here in this very large protein has led to the problems that we found in this baby, we thought. Now, what is beta-actin? It's a protein for your cytoskeleton, which means it's in every single cell in your body, and it's absolutely critical for the way your cell works. However, just because we found this mutation in the babies and not in the parents doesn't mean that it is the cause for the problems in these babies. So there are a variety of ways we can drill down on that and try and understand whether this is the case or not. First, there are now lots of databases of all the genome mutations that are out there, and software that allows you to predict how pathogenic these sorts of problems would be, depending on the effect on the protein structure. And indeed, those processes did suggest that it is likely to be pathogenic. A mutation in the ACTB gene is indeed consistent with a very rare condition called Barrett-Winter cerebrovascular syndrome. And part of that spectrum includes something called the Fryns-Efdimos syndrome. And the reason I put that up there is as a plug for Salim Efdimos, who was an amazing paediatrician who worked here for many years, and this syndrome is named after him, and he was an amazing dysmorphologist. When we had gone through the literature, obviously, in great depth beforehand, and in one case series, the identical mutation is reported once with a baby with Barrett-Winter cerebrovascular syndrome, and that patient did indeed have apple-peel bowel syndrome and high blood sugar levels. However, that baby did not have high drops and did not have laryngeal dysgenesis. So still not a complete match, but certainly highly suggestive. Other variants of the ACTB gene have been described in 2 patients who did have high drops and bowel abnormalities, but also didn't have the other abnormalities. So this is all highly suggestive that this is likely a pathogenic mutation. But for the benefit of patients in the future, we wanted to be sure, and so we confirmed this. This is Austin Gandy and his group in the School of Biological Sciences, who did this in collaboration with us, designed a pseudoheterozygote Saccharomyces cerevisiae strain. That's a yeast, brewer's yeast. So cloned one of these yeast strains with both the wild type of the ACTB gene and also a variant matching the variant we found in these babies. This was then incorporated into a haploid strain, and therefore the ACTB gene equivalent was heterozygote, whereas the remainder of the genome was haploid. And then they cultured this to see what the effect would be on the growth of this yeast. And indeed, you can see here, these are the wild type strains, which means the normal strains, and these are the strains that have the serine to leucine amino acid change put in them. You can see the growth is completely different here, and likewise in the orange here, these strains grew much less well. And remember, this is a cytoskeletal protein, absolutely critical for all cells. So from this, we conclude that indeed this is a pathogenic mutation that's now been added to the literature and to all the genome databases. And of course, the important message about this story is 2 things. First, we have a fascinating situation of genetically identical babies with different phenotype, telling us, as we do know already, but reaffirming very much that your genome is not actually everything that relates to how you are. There are other things at play, the epigenome and those sorts of changes. And secondly, showing us quite clearly that this sort of test, with a very rapid answer, can give you an answer to critically ill babies with complex abnormalities that we don't understand, that can really inform you about what is the best approach to take for these babies. We do know from the literature that babies with a mutation in this gene, Barrett-Winter syndrome or Fryns-Efdimos syndrome, do not have a good prognosis. It's a very, very serious condition. And if we were able to do this very early on, right at the beginning of these babies' journeys, we would have been able to have those conversations with the family and perhaps prevent the baby from having unnecessary surgery. As it was, it helped us obviously in framing the path for this baby going forward. So that's where I'm going to leave it and hand over to our next speaker.
So for many of us, when we hear the word healthcare, the first thing that comes into our minds is, how is it delivered? And currently, doctors rely on the standard protocols for diagnosis and treatment. And these are the standard protocols that are based on what works for the average New Zealander. And normally we focus on the most common causes of death and disability. And when you think about it, it has worked well for certain diseases such as diabetes. However, in some cases, there are people who still remain undiagnosed, which also limits their clinical care for these patients. But when we take a step back a bit and think about these little helpless creatures, 5 of them die in every 1,000 live births in New Zealand. And at least one of these deaths is due to an undiagnosed genetic disorder within the neonatal care unit. So this presents a very important opportunity for genetic testing, as Frank mentioned. And for genetic testing, that's basically looking at the changes in the DNA, and sometimes they're called mutations or variants, and they cannot be identified through DNA sequencing. So now that I mentioned about DNA sequencing, the cost has really dropped, and as you can see here, from the very first genome when it was published back in 2003, the cost was over $100 million per genome. And as you can see from the graph here, the cost has dramatically dropped to less than 1,000 per genome, and that is due to the rise of the different sequencing technologies over the years. And it's also predicted to even go lower, and my guess would be in 5 to 10 years, it might even be $10. And the most interesting thing is that the sequencing speed has also changed. From the very first genome, which took over 10 years, at the moment, we can do the sequencing within 24 hours, which is quite amazing. So our question is, can we harness this technology, DNA sequencing, in critical care? So based on that question and the dropping cost of sequencing, and also through our philanthropists who have generously donated towards the newborn genomics program, which is a facility we have set up at the Liggins Institute. And these philanthropies are the Denis Family Trust, Keliha Charitable Trust, and the Tautoko Trust. We have built this platform, and for us the idea is to empower clinicians and doctors with this critical genome sequencing information that can help to inform the clinical care of the critically ill babies. And I just want to emphasize that we are not doing this alone. This is a collaborative initiative involving different groups, specialists, clinicians, geneticists, and most importantly, the community. And to simply put, this is a community working to individualize diagnosis, predict outcomes, inform patient management, treatment and management. And at the moment, we've got the ethical approval as well as the local approval to proceed with this program. And these are the faces behind this initiative, led by who will be the principal investigator, Professor Justin O'Sullivan, and also Patrick, who is stationed at the hospital, who will be leading the clinical team. So looking at the sequencing platform, Currently, we have 2 Oxford nanopore technology machines, which we call P2 solos. And these machines are smaller than a loaf of bread. As you can see, they're really tiny. And each and every this P2 solo is connected to a high-spec gaming computer. And we're expecting more machines to come earlier next year. And each of these setup costs around 50,000 New Zealand dollars. So looking at how this DNA will be sequenced, this is real-time sequencing. So as I mentioned, those P2 solos, they have a certain kit. As you can see, it's very tiny. As you can see here. This kit contains poles within them. So when you extract the DNA from the blood of the critically ill children, it's loaded into this kit here. And what happens within the kit is that the DNA is loosened up to form single-stranded DNA, and it's pulled through these poles where electrical signal changes based on the DNA base that passes through the pole, whether it's A, T, C, or G, which are the building blocks of DNA, and the compute power of the gaming computer will be able to translate that and give you the whole sequence. One of the major advantages of this technology is that this is real-time sequencing, as I mentioned. And that means that you can run sequencing within a very short time, let's say 2 hours, 3 hours, and I'll show you the advantage of that. And that is when the clinicians need very rapid genetic information for the critically ill children. And in some cases, we can let it run for a few hours, even a few days. And this is when you want to, one, to increase the sequencing depth, and also when, in some cases, you have DNA, a sample that has low quantity of DNA. And this is really interesting because it's very adaptable. And that is something we are very excited about. So what we want to do is that we'll be sequencing, as Frank mentioned, the DNA of the sick child, the mom, and the dad. And this will help us identify whether there are changes in the DNA and help us identify whether there are rare genetic changes that could be causing the child's critical illness. And we have done our tests at the moment. And what we have found is that we can actually squeeze down the time since getting the blood from the hospital to the time we give a genetic result to the clinician. We can compress that time to 1 week, which is very, very important when you think about it. And especially for these critically ill children, when every minute, every second matters. So now that I've mentioned that we have already set up this platform, the next question is, how does it compare to the current diagnostic space? What is currently being done in the diagnostic laboratories? So to do that, we did benchmarking, and this was done according to international standards. And the purpose of benchmarking is to assess the ability of the sequencing analysis tools to provide reliable analysis compared to what is currently being done in the diagnostic space. And in genomic variant benchmarking, we normally say that if you cannot measure it, then you cannot improve it. So we started with genomes we purchased, and these are genomes that are the current gold standard genomes that have specific mutations, and they'll help you check whether your platform can pick up these mutations. And this benchmarking was performed using tools and standards. established by the Global Alliance for Genomics Health and Benchmarking team. So based on the benchmarking, we found that we can accurately identify single changes in the DNA with very high accuracy. And each and every dot you can see here that represents different benchmarking protocols. So just to contextualize, we can accurately identify a single dominant mutation with an accuracy of 99.8% in a gene called KCNQ2, which is potassium channel. And variants in this gene causes neonatal seizures. And imagine if you can identify this mutation at very early stage, then these kids can be put under anti-epileptic medication, which can help with managing their seizures. Also, looking at the benchmarking, some of these the small insertions and deletions, which are problematic even in other technologies, we have shown that we can identify these mutations with an accuracy of over 92%. And just to contextualize that, is that we can identify loss of function indels or insertion deletions in this gene called SLC19A3, which is a thiamine transporter. These mutations, loss of function mutations in this gene, causes built-in thiamine-responsive basal ganglia disease. And one of the most important things is that if you can identify this mutation immediately, the child is born, then early treatment can be administered. And that is basically using built-in and thiamine, and has been shown to improve the prognosis. So you can see there's the opportunity to save lives using this technology. And one of the most important things I would like to share, as I mentioned, during the adaptability of this sequencing technology, is that we can identify structural changes even at 2 hours. So looking at this plot, so this is just a plot showing chromosome 1 to 22 and the sex chromosomes, X and Y. And at 2 hours, we were able to generate 2 million reads. And each dot here at the center line indicates the reads. And also what this plot shows that everything that is within this midpoint here is 2 copies of the DNA. Importantly, we were able to identify within 2 hours, there's this 5 megabase copy number gain, which basically means there's an addition of an extra chromosome here in chromosome 18, which can also, for people who know about trisomy 21. And as Frank mentioned, microarray take quite a while, over 2 weeks. And for us, we are showing that using this technology, within 2 hours, we can really adapt and identify some of these large structural changes. Also, within this well-thought-out platform, we are harnessing the power of technology. And in this case, we are using artificial intelligence. And this is just to speed up the process of genome interpretation, genome annotation, and genome curation. And why this is important is because specialist curators, like Polona here, who we are lucky to have in our team, they are quite rare to find. And she will be spearheading the curation and reporting the clinically actionable mutations to the clinicians. And just to show the AI, the AI technology that we'll be using fabric genomics, which will help with the genomic interpretation and identifying the clinically actionable genetic mutations. And using this technology, they have shown that they can be able to identify with accuracy of 98% of important mutations that can be clinically actionable. And also in terms of incidental findings, we don't want to find anything that causes adult onset. And we have a well-laid plan for that because we have an ethical guideline based on that. So just to summarize, this is an initiative where we want to empower our doctors, clinicians, and our whānau. And this is to help them deal with over 400 diseases where there is available treatment. And we are really hoping for a national rollout for this program in late 2024. And just to emphasize, we want this to be an exemplar for rapid sequencing, rapid genetic testing for precision medicine here in Aotearoa and for our people. And as Frank has mentioned, in Australia, they have such an initiative that has been running for 5 years through the acute care genomics. And what they have shown is that once you identify a result of a genetic test, you can be able to change the clinical management of over 70% of these critically ill newborns, which means that is less hospitalization, less invasive tests, and the most important thing is that they will have access to life-saving medication. So to wrap up, and also to just emphasize, we are not doing this alone. There's a whole collaborative, a collaborative initiative spearheaded by Professor Justin O'Sullivan. We have co-investigators across in the clinical space, scientists, and we also have Maori governance. And I would like to thank our philanthropist for the generous donation which has made the program kickstart. And that's the Denis Family Trust, the Kelea Charitable Trust, and the Tautoko. And I would also like to thank you, the community. And with that, thank you so much.
Everyone, and thanks to Denis and Frank for really setting the scene for what I'm going to talk about tonight. And I'm excited to be here to hopefully provide you with some insight into some of the stages that follow diagnosis and some of the pioneering treatments that are emerging for rare genetic disorders. So, as you've already heard, for rare genetic disorders, actually achieving the diagnosis can be incredibly difficult, and it often takes many months or even years to actually find out what's causing an individual's disease. But as the sequencing costs are dramatically decreasing and technology is advancing, we're really starting to see this rapid diagnosis of these genetic diseases. So given that we now have the ability to actually diagnose individuals within a mere few hours, it's now more important than ever to consider some of the potential treatments for these very, very rare or even unique diseases. So in some cases, treatment options might be relatively simple. Let me introduce Jessica. So Jessica presented with a rare disorder that causes epilepsy and was affecting her movement and general development. However, repeated tests failed to diagnose her condition. And so scientists and clinicians together decided to take a blood sample from Jessica and sequence her whole genome. And when they compared Jessica's genome to the reference genome, they found 6.4 million differences. And okay, this might sound interesting, but trying to pinpoint a causal variant from millions of differences is extremely difficult. And just to give you some reference, if you were to compare my genome with one of yours, you'd probably find about 6 million differences. So Jessica's just like all of us. So then they took blood samples from Jessica's parents and compared Jessica's genetic makeup to her parents. And they identified just 67 differences that were present in Jessica and not her parents. And one of these mutations happened to be in the SLC2A1 gene. And this gene had already been linked to symptoms that were similar to Jessica's. And so they could now say that Jessica had GLUT1 deficiency syndrome. And this means that she doesn't produce enough of the protein, which transports glucose from the blood to the fluid around the brain. And this disease is thought to affect around 1 in 25,000 individuals, although several studies have reported a much lower prevalence, which perhaps indicates it often goes underdiagnosed, likely due to a lack of genetic testing. But luckily for Jessica, the treatment option for this syndrome is actually very simple. And she was put on a low carbohydrate diet, which is tailored specifically for individuals with this syndrome. And it reduces the number of seizures she gets dramatically, and therefore improves her quality of life quite significantly. And the genetic testing also provided peace of mind for her parents, as it showed that the genetic mutation wasn't inherited, but rather spontaneous, meaning that it provides peace of mind if they do decide to have more children. So in Jessica's case, genetic sequencing identified the cause of her disease fairly quickly, and changing her diet provided a very effective treatment. But for some individuals or families, researchers won't be able to clearly identify the cause of the disease right away. And for these families, researchers like us can continue looking for the cause through other methods. And even when a cause can be identified easily, the knowledge won't always suggest an immediate treatment or come in time to change patient care. But identifying the cause can bring peace of mind to the family or help unite families affected by similar conditions, and also inform family planning decisions. So as you've already heard a little bit from Frank and Denis, if we can provide a faster and more accurate diagnosis, this can facilitate the development of and access to precision treatments. And the different types of precision treatments are shown here in this figure here, and I'll now walk you through a couple of these pioneering treatments. So one group of diseases I want to talk about is those caused by RNA splicing errors. So RNA, or ribonucleic acid, is an essential component in translating our DNA into functional proteins that our body can use. However, within our DNA, there are lots of non-coding regions which aren't needed for making functional proteins, and this is where RNA splicing comes in. It's essentially like editing a recipe to remove any unnecessary parts, ensuring that the cell only uses the essential information to make the proteins that it needs. So if you imagine this is a string of DNA, normal RNA splicing would delete the introns, which are the little black parts in the middle, and keep only the red parts, which are the exons, and produce this message: the big house is at the end of the street, and that will create our working protein. But if RNA splicing goes wrong, we might end up with a message that looks a bit like this, and therefore creates a faulty protein. And so ultimately, these RNA splicing errors can lead to problems in the cell's ability to produce the right proteins, and they often lead to rare genetic diseases. So one example of a disease caused by an RNA splicing error is called spinal muscular atrophy, or SMA, and it's the #1 genetic cause of infant death. Spinal muscular atrophy is caused by too little of a protein called survival of motor neuron protein, or SMN, and too little of this protein causes the motor neurons to die and the muscles that they control to waste away, meaning that the individuals can no longer move them. And bear with me with the slide, it looks a little bit mad. But the SMN protein is usually formed from the SMN1 gene, here. But in patients with SMA, the SMN1 gene is completely missing or damaged beyond repair. So therefore, these patients are reliant on the backup SMN2 gene. However, although this SMN2 gene does also produce the SMN protein, there's an error that occurs. And so during RNA splicing with the SMN2 gene, the 7th exon is skipped. And so we end up with a message that looks like this. The big house is at the of the street. And it therefore produces a faulty protein. And it's therefore a very poor backup of the SMN1 gene, as it produces very small amounts of the actual working SMN protein. But you might be thinking, if the SMN1 and the SMN2 gene are so similar, is there a way to repair SMN2 to make it work just like SMN1? So this treatment here, called Spinraza, is a new treatment that enables SMA patients to actually make more SMN protein from SMN2. And the treatment is a short sequence of RNA that binds just after the exon 7 and prevents exon 7 from being skipped, meaning the functional full-length SMN protein can be formed and reverse the symptoms in these patients. And it was decided just last year by whānau that they would fund Spinraza for SMA here in New Zealand for individuals who meet the criteria. And so this form of treatment is known as an antisense oligonucleotide, or ASO. And antisense oligonucleotides can essentially be developed for any condition where an RNA splicing error is the cause. And so this brings me on to a more personalized case of an ASO. And this is a treatment called Millicin, which was developed for a small girl called Myla. Myla was born perfectly healthy, but at the age of 2, she started to develop symptoms, including speech impairment and abnormal movements. She was eventually diagnosed with Batten disease, a rare neurodegenerative disease, which causes children to die at a young age. And similar to SMA, an RNA splicing error was the cause of Myla's disease, leading to a faulty MFSD8 protein. So together with Myla's parents, scientists at Boston Children's Hospital decided to launch an N of 1 trial and developed Millicin, this ASO drug that could restore the function of the faulty protein in Myla. Now, unfortunately for Myla, the process was quite slow and the treatment ended up being too late. But this pipeline does really highlight the potential for the development of these treatments for completely unique or rare diseases. And this timeline here shows the time it took to develop Millicin, from the time of diagnosis right the way through to the time the drug was in production. So as you can see here, it took around 7 to 8 months to actually identify the cause of Myla's disease. But as you've already heard from Denis in particular, if we can implement this rapid diagnosis platform, then we can likely identify defects in or genetic mutations that are causing similar diseases, hopefully in 7 days, or at least a lot shorter than 7 to 8 months. And as you can see, if we can speed up the time to diagnosis, then drug development can start much sooner, hopefully leading to much better prospects for these individuals with rare genetic conditions. So now I'm going to change track slightly to a different disease and a different set of treatments. So metachromatic leukodystrophy, or MLD, is a rare genetic disease that affects around 1 in 100,000 live births, and it affects the body's metabolic syndrome. And it's caused by mutations in the ARSA gene, and it causes damage to the nervous system and causes a buildup of unwanted fats in the brain, among other organs. And so, although individuals can live with this condition, infants with infantile MLD typically die by the age of 5. But we can fix these mutations using a technology known as ex vivo gene therapy. So Leo here has MLD and has undergone this ex vivo gene therapy to fix his condition. So firstly, clinicians took a blood sample from Leo and they isolated the stem cells from his blood using a special machine. And stem cells are special because they are essentially a blank slate that have the potential to become any cell that Leo requires. And the isolated stem cells are then taken to the lab, where the working copy of the gene can be delivered using a lentiviral vector into the cells. And whilst this is happening, Leo receives chemotherapy to help make his body ready to receive his new improved stem cells, essentially making his body a bit like a blank slate to receive the new working gene. So Leo then receives an infusion of the new stem cells, and once they're back in the body, they can divide and make copies of themselves so that the faulty gene can be replaced with this working gene. So in Leo's case, the faulty gene activity has been restored back to normal, meaning his cognitive function and motor development has been fully restored. And this is a treatment that's being done in some other countries, but not yet here as far as I'm aware. So the treatments I've spoken about so far are already fairly well established, or have at least been trialled for rare genetic diseases. However, there are some other types of treatments that are already being trialled for more common diseases, but they have the potential to one day be adapted for some of the more rare diseases. So this is a little bit of Justin being channeled through me, but microbiome therapy is one example. And at present, therapies that modulate the composition of the microbiome are often used for the treatment of common metabolic or gut diseases, such as IBD. However, it is possible that in the future, some of these rare genetic diseases that play havoc with the gut may be treatable with microbiome therapy. And some of you may know that researchers here at the Liggins have been exploring the potential utility of fecal microbiome transplants for more common diseases, such as autism and anorexia nervosa. But there is the potential that, given that we have already got this platform set up, that perhaps in the future these transplants could be developed in a more personalised way for the treatment of some of these rare genetic disorders. And another very novel and exciting technology is precision gene editing. So this technology, as Frank alluded to earlier, uses CRISPR-Cas9 to edit a single faulty letter or mistake in our DNA and replace it with the correct letter to restore normal function. And precision gene editing has recently been trialled in humans to treat dangerously high cholesterol levels. And if we compare this to the ex vivo gene therapy that I described for Leo earlier with MLD, this treatment can be done in vivo, meaning that patients just have to receive the treatment via an injection. And also very recently, and some of you may have seen this in the news, a gene editing therapy has been approved in the UK with the promise to cure sickle cell anemia and beta thalassemia by correcting the genetic mutation responsible for causing the disease. And these treatments have been called one and done therapies, as one treatment in theory is enough to cure or reverse the disease. And so this is particularly relevant for rare genetic diseases, as many of them, such as the case that Frank described, are caused by single faulty letters in our DNA, meaning that this form of gene editing therapy has great potential for developing future treatments for these diseases. And at the moment, a major issue for these pioneering treatments is the cost, often being dubbed as $1 million treatments. However, we're confident that as the development of these therapies becomes more common and efficient, the cost is likely to dramatically decrease, just as we have seen for other technologies like sequencing that Denis showed us just earlier. And although exciting, we do have to remember that precision gene editing does change an individual's genome forever, meaning that the associated ethical and safety issues and long-term effects do really need to be considered. So as you can hopefully see, diagnosis and treatment of rare genetic diseases is advancing rapidly, and the possibilities keep expanding as technologies continue to improve. And as is happening in other countries, such as the UK, genetic analysis are likely going to become part of the standard of care in many instances within the healthcare. And we really hope that our platform can, to some extent, help to drive this. Ultimately, the more we understand about the root cause of the disease, the better the treatment is going to be, especially when considering precision medicine. Thank you for listening, and I'm sure Denis and Frank and I will be keen to discuss over some drinks and nibbles outside.